Hi everyone. Today I wanted to talk to you about developability of antibody-based therapeutics and some exciting developments in this field. First of all, let me begin with saying what is developability and why it is important. Really, as a pioneer in the field, I would think that developability is our ability to develop a newly discovered drug candidate as a product and as also through the clinical trials and get approved and really become available in the market. So it's the translation of antibody drug candidate into an antibody drug product that has been approved and is available in the market. That's developability. It is much broader in the scope and much more complex than what we typically think of it merely in terms of manufacturability or biophysical attributes. The importance of developability lies in the fact that how can we improve the productivity of antibody-based drug discovery and drug development cycles, and how prepaying for developability issues in the drug discovery can actually set us up during product and clinical development, and therefore bring more medicines to the patients faster and at lower costs. Essentially, therefore, for this to happen, and a given antibody drug candidate needs to, of course, bind its intended target, but also it needs to be safe, effective, and easily manufacturable, and needs to be stable during storage, shipping, and should have a good long enough shelf life in the pharmacy's shelf. So essentially, all of this is developability. As a drug developer, I would say drug development is very, very complicated process and very costly and time consuming as well. And the hurdles that developability poses in this journey of product and clinical development can even sometimes kill the program. However, fortunately for us, there are a few intrinsic properties, intrinsic biophysical properties, intrinsic sequence and structural properties that we look for, and because of which we think that probably if those properties are good, the antibody has a good developability, and therefore it has a better chance of succeeding. And some of these properties are biophysical. For example, stability, self-association, as well as polyreactivity. And some of them are essentially sequence and structure based. So for example, humanness, presence of physicochemical liabilities in the CDRs, immune epitopes, the distribution of charge and hydrophobic patches on the molecular surface, all of these, and the PI, all of these actually contribute towards that developability. And having a good developability essentially means we have a favorable biophysical attributes as well as favorable sequence and structural attributes in a given antibody-based therapeutic candidate. Really, then the question is how to make antibodies developable, or how to make antibody candidates developable. And this is best done during the drug discovery stages rather than in drug development, because at the drug development, the sequence of the antibody candidate is already frozen. It's already logged in. So we cannot change or optimize antibody sequence. But at the drug discovery stages, we can triage hits as well as optimize the lead candidates for function as well as developability. And there are two ways. One is the lab way, really create mutations, study the impact of each mutation, and keep the ones that are favorable and leave out the others. There are also in silico way, where we actually study different attributes and then try to optimize them for developability. One more thing that I would like to add here is that As a drug discoverer, I always found it useful to engage my drug development colleagues early in the drug discovery processes so that I am already setting up uh, for their success and, of course, our collective success for drug discovery and drug development projects. And now, in thinking about in silico ways or how we can do computationally, how can we improve or optimize lead candidates for developability, there are sequence liability removers, aggregation prone regions, immune response. Those are the engineering techniques that are antibody engineering techniques that we are familiar with it. But we are also familiar with the profiling method. And because the data on antibody therapeutics that have been already approved is still small, profiling methods can actually provide us some guidance based on what we see in approved biotherapeutics or clinical stage biotherapeutics. And based on that, we can see how likely our drug candidate is to reach in the clinic or get approved. Of course, these are not guarantees. These are more of a mathematical estimations. So some of the profile-based methods, I think the earliest base was called Developability Index. That was from Bernhard Raut's group. And then Therapeutic Antibody Profiler from Charlotte Dean's group basically introduced the concept of comparing the candidates, or the hits or lead candidates, with respect to the reference molecules that are the molecules or the antibodies that are in the clinic or have been approved. And then later on, we ourselves had created a medicine likeness profile. Another thing that in the recent years that we are seeing is that how are we able to predict the outcome of biophysical assays from amino acid sequence and structural properties of the antibody variable regions, or maybe full-length antibodies from candidates. So there have been a few studies. One of them collected lots of drug developability data set and lots of methods and just try to create a machine learning model to be able to predict based on the sequence and structure the outcome of those developability experiments. And then more recently, there was the Ginkgo competition where we had a set of test antibodies and the training antibodies, the biophysical data, and was made available to everyone. And then people try to predict individual biophysical parameters such as Higgs retention time, AC sense, and titer, polyreactivity, all of these things, biophysical measures based on the antibody sequence and structure databases. The results from these studies and this competition really showed that we are ways away from being able to accurately predict individual developability descriptors or individual developability biophysical measures from the sequence and structural properties of the antibodies. So in this way, what can we do? What are the other alternative ways we can think about? And I think one thing to mention over here is that developability, at least from a drug developer's perspective, is more of an optimization problem rather than a maximization problem of one particular biophysical parameter. We do see from these studies that individual descriptors have some weak correlations with the experimental values, and they have some predictive value there, but we are not completely there. And in this process, there are also a welcome development, which is essentially using the protein language models for developability prediction. And when we think about in terms of the protein language models and how do we go about predicting developability, the two most common talking points are essentially that we don't have enough data, or we need to do more, we need more data to fine tune our language models. Um, in natural antibody, we are always thinking about how do we serve our customers better. So we have been historically collecting lots of public domain as well as generating in-house the data to train our own internal AI tools and systems and to fine-tune them. Also, I want to then now talk about really Apollo. It's a co-scientist AI agent. Essentially, it has the data, it has the fine-tunings and all done, but also more importantly is that it is able to converse with the experts in the field and is able to incorporate those perspectives. And therefore, for Apollo, what we have really done is that we have combined our AI models, our in-house and open source data on antibody sequences, structures, and developability, and then contextualized this LLM in terms of antibody engineering or antibody design. And therefore, when this LLM or this agentic AI that we call it as Apollo, when it suggests certain mutations at the lead optimization sequences, for example, it not only tells you what mutations to do, but also adds the justifications to it. So it's a more of an expert-driven or expert antibody lead optimization antibody process. And I hope you would like the agentic AI that we are building. And it, of course, it will be available through Digital Biologics. And we'll continue to further work on it and keep on improving it. At this time, I just wanted to inform you about this development. And thank you so much for your attention. And feel free to reach out to us if you have any questions. Thank you.